Good morning. I am delighted to have the opportunity to welcome all of you here at the 10th anniversary CIMeC Welcome and Doctoral Student Day. In fact, this year, for the very first time, we have meshed these two events. We have the opportunity to welcome the new welcome students from the 32nd cycle, and also at the same time, they will have the opportunity to listen to some of our colleagues, some of other PhD students coming from different stages at the PhD course, and to listen four of these students, and also to see what they are doing, what they are working on in the poster session. There will be also time for questions, few relevant and very important questions for the speakers, and also there will be time for a prize giving. Please don't forget for PIs and faculty members and the postdoc to vote for the 3 best presentations. They could be both oral or a poster presentation. You receive a sheet, and in this you have to vote free person, free presentation, and to give this paper to or Veronica Mazza, or to put in the box, Veronica Mazza is there, to put in the box in the poster area downstairs, just before 1:30 p.m. Well, the day will be like this. So now we have from 9 to 11 the oral session. After this time, we will have the poster session downstairs and a well-deserved coffee break. After the coffee break, we will have a lunch, a welcome lunch for all the new students and all the past students. After the lunch, there will be the welcome speech from the CIMeC representative, and after that, the colloquium from our guest. At the end, when you will be really tired, you will have the opportunity to visit one important feature of the PhD life, so the coffee and the break. Very important, remember. So let's go deeper in to make vivid your new experience, to start the new life as a PhD and see and taste how it will work, inviting the first speaker for today. And the first speaker will be Aidas Aglinskas. Oh. He works with Scott Farrell in the FAIR Lab, wonderful name. His main topic research is the representation of abstract semantic concept in the brain. He just started the second year. Okay? And he has a nice background both in psychology and in neuroimaging. Welcome, Aidas.
Good morning. Can everyone hear me okay? Okay. I want to thank the committee for granting me this opportunity. Today I'm very excited to talk to you about my research, So for my PhD, I work on a project called CRASC, which is an investigation into how the brain represents abstract semantic information. And for us as social animals, there's no better source of semantic information than other people. Because every time we see a familiar face, we are able to recall facts about that person. We are flooded with memories of times that we spent with that person. We're able to recall their name. We know, we have an idea about how attractive we find that person. And there's so much more. And another thing that makes faces great is they also have been extensively studied. Previous research has identified a person perception network that gets activated every time we see and think about familiar people in our lives. And this network is comprised of a core system involved in perceptual features, and it involves regions such as the fusiform and occipital face areas, as well as the posterior superior temporal sulcus. There's also the extended system, a set of regions that are also implicated, that are also activated when we think about familiar people. And the regions of the extended system themselves have been implicated in a wide variety of tasks, from episodic memory to default mode and more. And there are also some new arrivals, such as the recently discovered, not too long discovered, anterior face patch, whose role in the system is still not clear. Now, these regions had been studied in in isolation extensively. But what we wanted to see is something very different. We want to see how these regions behave as a network. What is the pattern across these regions when we think about a person? And what happens, how does this pattern change when we think about different aspects of people? So really take this the systems level approach. And we knew that to address this question, a standard approach of having a couple of tasks, contrasting them in a univariate analysis, looking at a brain map just simply wouldn't do. So what we've developed is an experiment where we show our participants a face of a famous person, and then we ask them what is that person's name. And then we ask them what is their job. How attractive do they find this person? How friendly? How trustworthy would they rate this person? And we keep on asking questions. How familiar do you find this person? How common is that person's name for them? How many facts can you recall about the person? What is the first memory you have associated with a person? And how distinctive is their face? So for every face that we present, we ask 10 different questions of our participants. Now, this is a peculiar approach, and why do we do it? Well, as our participants are thinking about a name of a celebrity, we're able to record the brain response to that cognitive dimension. And as they think about their job, we obtain another response. And once we obtain a response for all of the 10 tasks, we are able to capture a response profile for the regions involved in person perception. So we had 20 participants go through this experiment, and we collected their brain data. And what do we do with it? Well, at its most fundamental, we can summarize. So grouping together similar tasks and collapsing across regions of the opposite hemispheres, we arrive at a cognitive fingerprint, which allows us to not only see the magnitude of activity, but put it in context. And that is already giving us some fresh perspectives on things. So for example, consider the ATL. Previous research has implicated ATL as being critical in proper naming. And in context, what we see is that while the ATL is activated for naming tasks, when we look at it in context, we see that that's not its highest preference. The ATL prefers social tasks more than it prefers nominal tasks. And other regions, such as the IFG, prefer nominal tasks more than the ATL does. So by looking not only at activity, but also the activity's context, we're able to get a deeper understanding even what this individual regions do. So this is a cognitive fingerprint that's summarizing the data. But why summarize the data when we can harness the complexity? Because the reason we collected, the reason we have the 10 tasks is to make use of them. So what we do is we collect the response profiles for every region involved in person perception, and then we look for similarities between these patterns. So what it allows us to do is to employ some of the multivariate methods that have been very successful at explaining some of the complex behaviors that the brain is engaged in. So for example, one thing we can do is we can not only see what is the activity when person is thinking about a person's name, but we can also relate it to other activity. So now we have this cognitive taxonomy of how different tasks relate to each other. But the most important question we wanted to answer is how do all of these regions function together as a network? So this is the person perception network in its canonical form with the core system, extended system, and the unknown face patch. And if we look for patterns of similarity to find regions that communicate, that work together when we are looking at a familiar face, we observe something different. So we see that the two regions of the core network communicate very actively with two frontal regions, the IFG and the orbitals. Their response profiles are very similar, and they act in unison, so they're a functional unit together. We see that regions implicated in the default mode preserve their relationship even in task, And when we think about different aspects of the face, they are communicating to each other and performing in unison. And also we can address the face patch. So face patch has a very similar response profile to the amygdala. So they also tackle problems and tackle tasks as a functional unit. So, but also not only do we see which regions go to which other regions, we also see the relationships between them. So it's a complex network. Sorry, thank you. So it's a complex network with complex relationships, but when we look into it, we see that, for example, right posterior STS, of course, its closest functional friends are other regions of the default mode. But it also actively communicates, because it's part of the core system, it also actively communicates with the other functional subunit of the other two core regions and the two frontal regions. So this is very interesting. This is a novel insight. So in the person perception network, there are three distinct clusters of regions that perform similarly and couple together to address, to enable us to do everything that we do when we see a familiar face, to be able to extract all the semantic information. We wanted to take a step further and see how reliable are these clusters. So if we wanted to address the question, if we were to collect 20 more subjects, would we observe the same clustering? And what we did is we bootstrapped across subjects, and we see that the clusters are very reliable, with the exception of the right posterior STS, which goes just as likely with the core frontal subnetwork as with its default mode buddies. So to summarize, we've approached person perception from a new angle. We focus on pattern of activity instead of magnitude of activity. And we think that allows us to look at data in a fresh way. So not only we see what this region do in this particular task, but we can also relate it to what it does, how it relates to other cognitive dimensions. So also looking at patterns of similarity, seeing how does the network come together under load, allows us to probe deeper and to actually see the task-dependent organization of the network. We see deeper than we did previously into how the network performs what it does. We see that default mode provides a specific contribution, as well as the other two core regions of the network coupling with the frontal counterparts. And we see that anterior face patch, which role wasn't that clear, might actually be implicated in the extended system. So for future direction, since this approach has given us new insights into person perception, we are thinking about we're going to expand it to wider semantic categories, because those have even more complexity. So, and this approach of harnessing complexity in our benefit, I think, is going to have even more great insights in wider semantic networks. Additionally, once we capture the network in action, once we have a model of how that looks like, we can transfer it between people and as well as different populations to see what is different in networks, this network's activity. For example, if we look at an autistic brain or other brains in general. So I think this is a powerful approach. And yeah, thank you very much. I'm happy to answer any questions you have.
Thank you, Aidas. We have time for question.
Yes.
So maybe it's a matter of definition, but I don't understand what you mean with person perception. So let me put it differently. What's the difference between person perception and face recognition?
Face recognition, those are actually two very related concepts, because a person has to have a face, and
you use face only, right?
Yes, we use face only, but the difference between looking at a face and thinking about a person is the depth of semantic access. So usually when we say face recognition, we count familiar and unfamiliar people, but there's something special that happens when we think about people that we know deeply. So there's a much richer content of semantic information that we can access.
Well, you don't know. I mean, when I see a face, I can go deeper and deeper. It's just that if you ask me just to say whether it's a familiar or unfamiliar, then I will just comply with the instructions.
Yes, but for the unfamiliar faces, we simply don't have that semantic information associated with it. So that's the difference between a face and a person in our lives.
Do you actually check for the communication between those nodes, or is it just based on the similarity of the activity? Because you keep saying that they communicate extensively, but is there actual communication between them? The way we define communication is we correlate the response profiles, and we arrive at a similarity matrices. So really, so if the two regions respond to similar tasks, they have to be communicating. It's very unlikely that just happens by chance.
Yeah, so I was wondering, what do you, actually you assume there's these networks related to face recognition, person perception, and all that. What can you infer about those networks? Like cognitively speaking, you just have now we're talking at brain level, can we infer something about abilities or somehow features of perception and recognition by just looking at the networks?
Can we infer something from the network's activity, something about cognitions? Of course. So the advantage of looking at patterns is once we go as an aside from the regional connectivity, we also have the connectivity profiles of the different tasks. So we can see how this network in general, or if you want, how this particular person treats the different tasks. So some tasks are very, very different. Some tasks are quite similar in their activation profiles, and I think that can give us an insight into how the particular person is structured cognitively. Does that answer that? Thank you.
Yes. Just to be clear, so you can pin down these semantic connections to different networks. So you can say connection between the FC and amygdala is more related to these features rather than other features. Can you say that?
No, actually, it's across the whole pattern. So we want to see how are they connected, how are they overall connected. So how to say that? So they have to communicate all of the information. It's hard to pinpoint. It's hard to pinpoint that they only focus on this one tasks.
Okay.
Yeah, we haven't subdivided it into particular tasks so far. No.
Okay, thank you. If we have time, we can ask some more questions to all of the four speakers. Okay. Thank you once again.
Thank you.
Our next speaker is Giulia Malfatti. She works with Luca Turella. She works in the Motor Control Group Lab, of course, about motor system and action. She just started the third year, I suppose, the Second, wow, she just started the second year. She has a nice background also in psychology and the cognitive neuroscience. Welcome, Giulia.
Can you hear me? Yeah, okay, cool. So good morning, and thank you to the committee for this opportunity. I'm Giulia Malfatti, and today I'm going to present you the project that I worked on so far in my first year of PhD. So before I go in details with the study itself, I just want to tell you why I think it's important to study motor control, and hand movement in particular. So in our daily life, we strongly rely on our hands in order to interact with the environments we live in. For example, driving, drinking, or using our mobile phones are all activities that we can easily perform thanks to our hands. The loss of this ability can have dramatic consequences. A successful interaction with the surrounding world needs our brain to process complex and different informations. In the classical model of Milner and Goodale, there are two visual pathways regulating our interaction with the world. The dorsal, which is responsible for vision for perception, so we are considering the planning and the online control of an action, and it runs from the occipital lobe through the parietal, and it reaches the frontal regions, and the ventral stream, responsible for vision for perception. So we are talking about the identification and recognition of objects. This runs from the occipital lobe through the temporal, and it reaches the frontal regions. Within the dorsal stream, the two pathways seem to be mainly involved in the hand actions. So the dorsomedial and the dorsolateral pathways. It was thought that these two pathways computed different information about hand movement. The dorsal was considered to be more involved in the online control of an action, while the dorsolateral was considered to be more involved in the visual motor transformation for goal-related actions. Only recently, some monkey studies and very few human studies suggested that these two pathways are not functionally segregated as thought. They are both actually recruited by the same tasks, suggesting an interaction between the two. So what it has not been explored yet is what type of information are exchanged and which areas of the two pathways are involved in this interaction. And here we arrive at the study itself. So in this study, we wanted to investigate the causal involvement of cortical regions within the two pathways, and we wanted to identify the connectivity profiles within and between the two pathways. We did it using a combined approach of methodologies, and we used transcranial magnetic stimulation and the functional magnetic resonance imaging. I will go through the first issue first. So, as I said, we used TMS, and the area that we stimulated is the superior parietal occipital cortex, SPOC. This area is located in the posterior part of the dorsomedial pathway, and some monkey studies showed that it seems to communicate with the dorsolateral pathway. So we were curious to see if by perturbing the activity of SPOC, we could cause an effect in the connected areas. Moreover, in a previous study run in our lab by Simona Monaco, SPOC happened to be extremely activated during the planning and the execution phase of a grasping action, mainly when participants could see the object they were going to grasp. As we used a very similar task as the one used by Simona in her experiment, we thought to be nice to use the same an area that we already knew to be activated by the task. So in this study, we use a 2x2 factorial design, where the first factor is represented by the TMS stimulation. So we use a repetitive offline TMS stimulation of 1 Hertz over SPOC at 100% of the motor threshold of each participant, or a sham stimulation over the same site. The second factor is represented by the visual feedback each participant could have about the object they were going to grasp. So we have a visual condition where participants could see the object, and a blind condition where participants had to keep their eyes closed. So as I said, TMS had two levels, an offline repetitive TMS stimulation and a sham stimulation. The TMS preparation consisted in the neuronavigation of each participant's head in order to identify the right position of SPOC to be stimulated. And we measure the motor threshold of each participant in order to set the right intensity for the stimulation. The second factor, the visual feedback, is related with the MR setup and the task. The task consisted in a delayed grasping task. Participants had to lay in the scanner with their head tilted so they could directly see the object they were going to grasp. And the object was a fiber optic button fastened on a plexiglass support placed on participants' hips. They had to plan and then to grasp the object with their right hand, either with their eyes opened or closed. At the beginning of every trial, each participant had to keep their eyes closed. Then an auditory cue instructed them if they have to keep them closed or if they had to open them. After 10 seconds of delay, which represent our planning phase, so the time windows we are actually considering in our analysis, another auditory cue instructed them to start to perform the action. After 2.5 seconds, another cue told them to stop the action, close their eyes again, go back to the home position, and waiting for the next trial to start. Each session consisted in 4 runs. 2 were performed after 20 minutes of offline repetitive TMS stimulation, and 2 were performed after 20 minutes of sham stimulation. The order of the runs was counterbalanced between subjects. The analysis that we performed on the planning phase of our task are a univariate analysis, a multivariate pattern analysis, and a dynamic causal modeling analysis. So the univariate analysis was used in order to identify the prehension network recruited by the task. Both dorsomedial and dorsolateral pathways are recruited during the planning phase, particularly for visual condition. However, we decided to adopt MVPA approach to evaluate if TMS was inducing modification at the level of representational content within the brain. We performed a searchlight MVPA, and we asked the classifier to distinguish between the visual and the blind condition. We did this for the sham runs and the TMS runs separately. So our idea was that the TMS perturbation might cause an effect within the stimulated areas and in the connected regions at the level of representational content. So to test that, we subtracted the decoding accuracy of the sham runs, of the TMS runs, from the decoding accuracy of the sham runs. So if the TMS does not affect the representational pattern for the visual and the blind condition, then the results of this subtraction should be equal to 0. What you see on this mesh is the map of the difference between the decoding accuracy of the sham and TMS runs. So as you can see, there are some areas in the brain that, that changed their representational pattern when we stimulate SPOC with the TMS. These areas are mainly located in the left hemisphere, and they are the ventral premotor area and some big clusters, including the intraparietal sulcus and the supramarginal gyrus. All these areas are located in the dorsolateral pathway. So what I think it's interesting to remark about these results is that we are stimulating an area in the dorsomedial pathway, but we are causing an effect in the dorsolateral pathway. So these results goes in support to the idea that at some point dorsomedial and dorsolateral pathways communicate. However, with this analysis, we can't say where in the brain the interaction occurs. So we now need to explore the second issue. And we need to identify the connectivity profiles within and between the two pathways. To do so, we use the dynamic causal modeling analysis. So the nodes that we selected for this analysis are SPOC, as it's the target of our TMS stimulation, and we needed to include it in the analysis, and 3 more areas taken from the literature as part of the prehension network. They are the anterior intraparietal area, the ventral promoter area, and the dorsal promoter area. So we have SPOC and PMD for the dorsomedial, and AIP and PMV for the dorsolateral pathways. So with DCM analysis, we can investigate the connectivity between the nodes selected. And the aim of DCM is to make inferences about effective connections and their changes in different experimental conditions. In the intrinsic connection, we are considering the overall experiment. In our case, we are considering the planning phase entirely, including both sham and TMS runs, visual and blind conditions. Then we wanted to see how different conditions can modulate the connectivity between the areas, and the modulatory effects that we tested are the visual condition, the blind condition, and the TMS effects in both visual and sham conditions separately. So today, I'm going to present you only the results for the visual conditions. So DCM results show that the planning phase of our task significantly recruits all the connections of our model. In red, you can see a positive and significative connectivity, and in green, negative and significative connectivity. So the photic label that you can see on the upper right corner represents the nodes where our information enters our network. About the modulation, in the first pink square, you can see that the visual condition increases the connectivity of the entire network, except for the backward connections in the dorsolateral pathways, which is reduced, and the connection between the dorsolateral and dorsomedial pathway that is not significantly altered. In the light blue squared on the right, you can see that the TMS in the visual condition positively highlights the connectivity between the posterior nodes of the dorsomedial and the dorsolateral pathway, and the connection within the dorsolateral pathway. So in conclusion, DCM analysis showed that the visual feedback modulates both dorsomedial and dorsolateral pathway, and that TMS in the visual condition particularly affect the posterior nodes, the connectivity between the posterior nodes of the dorsomedial and the dorsolateral pathway, and within the dorsolateral pathway. So, and here we arrive at the very last slide. And what I think it's interesting to keep in mind about this study is that both our analysis, so MVPA and DCM analysis, confirm or supports at least the hypothesis of communication between the two dorsal streams. In particular, we show that there is a recruitment of the dorsolateral pathway during the planning phase of a grasping action when we manipulate the activity in an area of the dorsomedial pathway. So thank you for your attention.
Questions?
I apologize, perhaps I missed it. Did you try a Bayesian model selection for the DCM, and did it select the pattern you had, or did you a priori pick that?
No, we didn't, because this was our a priori hypothesis. So based on the literature, we used a model which supported the idea that all these areas are connected. So this was our a priori hypothesis, and we didn't try any different models.
Okay, thank you.
If you have other questions, we can ask them at the end of the talk. Thank you. Our next speaker is Mariagrazia Popeo. She works with Angelo Bifone in the BrainNet lab. Her main research area is the study of the functional connectivity with the optical imaging. She just started also the second year, and she has a background in physics. Thank you, Mariagrazia.
Okay. Hi, everyone. Can you hear me? Okay. I'm very excited to present my PhD project today. I'm Mariagrazia Popeo, and I'm working in the BrainNet lab, supervised by Angelo Bifone. And before I start talking directly about my project, I just want to spend a few words to introduce you the concept of functional connectivity, because it will cover a large part of my PhD project, of my work. So we can define our brain as a network, and a very efficient network, to be precise. And this network is characterized by different brain regions. Each of these brain regions has its own role, its own task, but they continuously communicate with each other. So we can measure the level of the communication between brain regions. And we can define the resting state functional connectivity as the temporal correlation of spontaneous low-frequency fluctuation recorded during rest. So it is likely to be a key rule in the comprehension of complex cognitive systems. And resting state functional connectivity is able to reveal the organization, the structure of a group of brain regions that we can refer to as resting state networks. These resting state networks are spatially spatially separated, but functionally linked brain regions. And they are characterized by precise spatial patterns. And resting state functional connectivity can be considered as a marker for its potential role to neuropsychiatric diseases or developmental disorders such as autism or hyperactivity disorders. So it could be very interesting to study the functional connectivity in the developing brain, but in particular way in the infant brain. It could be at the same time a unique opportunity, but also a challenging work. And the aim of my project is exactly the investigation of the inception and development of functional connectivity in newborn infants. We want to understand the principles that govern the organization of the brain at a very early stage of life. So we will use the functional near-infrared spectroscopy to measure resting state networks. In particular way, we will focus on one precise resting state network, the default mode network, because we know very little about its emergence. And in order to be sure of covering the right areas of the brain of interest, we have worked and we are working on the optimization of the probe design. This is a very important step in our work because it will allow us to get close to a first description of the phenomenon. And as last step, we will analyze data and we will give a broader description of the functional connectivity using also the graph theoretical approach. So let me give you a brief background of the technique that we are using for measurement and how it works. So we know that functional state of tissue can influence the optical properties, and our brain, the human brain, undergoes a series of physiological changes as it responds to environmental stimuli. So functional optical imaging uses light in the near-infrared window, in the near-infrared range, to study the changes of these physiological changes. In particular way, due to the fact that oxygenated and deoxygenated hemoglobin have characteristic optical properties, we can study the changes in the concentration of these molecules during neurovascular coupling. And during acquisition, all sources are fixed on a cap in order to be adjacent to the scalp. So in this way, near-infrared light can penetrate the scalp, the cerebrospinal fluid until reaching the cerebral layers of the brain. And photons can penetrate tissue, and when penetrate tissue, they can be scattered or absorbed by it. But we know that a relatively predictable quantity of photons follow this characteristic banana-shaped path to skin. So we can use a photodetector to detect these kind of photons. We put photon detectors a few centimeters distance from the source, and we define channel, the pair of source and detector. And we define probe, all the set of channels, so all the set of sources and detector. Usually probe, as you can observe as you can see in this picture taken from a previous experiment of Matteo Caffini, probe is carried out in a symmetrical way. So we have in each hemisphere the same geometry and the same number of sources and detector. And if we manage and maintain the sources adjacent to the scalp, we could have a lower risk of movement artifact. But why should we choose functional NIRS? Because we have a lot of other neuroimaging available, in particular way in adults, but we cannot straightforwardly extend this technique to infant. For example, we can consider the fMRI or the MEG. They necessitate the subject to lay still in the scanner for an extended period of time. Or PET is highly invasive due to the use of radio tracer. Conversely, functional NIRS is totally non-invasive. We can avoid to use a sedation with baby in functional NIRS. It has an acceptable spatial and temporal resolution trade-off, but it presents a series of important advantages, practical advantages. I'm talking about the portability of the technique, the possibility to realize a bedside measurement, silent bedside measurement, the low degree of tolerance needed from infant, and we have to consider also the fact that infants are good subject for functional years due to their thinner skull or minimal hair or a smaller height size. But we have to consider also the limitation of the technique because most devices have a limited number of channels. In our case, we have 20 channels, and we will use them to investigate a particular resting state, the default mode network, also referred to as DMN. So there is a growing, this is one of the most studied and observed network in adults. And there is a growing interest in understanding its role in the brain functional architecture and in understanding how it is affected by disease. But unfortunately, to date, the majority of studies on default mode network focuses on adults. So we know very little about its emergence. We don't have information about at what stage of development it emerges. So for this reason, it is important to study the default mode network in particular way in infant. Because I just want to do a small consideration on the importance of study infant newborn brain. In fact, we cannot consider the infant brain as a miniature version of the adult brain. It is a continuously self-consistent changing system that forms a connection between different brain regions via short range or long range. So it is not correct to extend the information that we have of the default mode network in adult straightforwardly to infant. And so the main driving question on my study is the following one. Is it possible to detect the default mode network in newborn? Because to date, the literature seems to show discordant results. So I just want to show you some results from different studies. For example, in this case, we have a study on early preterm and term equivalent infant. And they seem to find a network that resembles in a certain way a complete default mode network only in term infant. This could be a very interesting positive result because it means that we are able to detect default mode network before the acquisition of cognitive processes. But on the other hand, other studies, like this one of Franson's, failed in finding a complete default mode network. So they found a network that shows only in a partial way the same characteristic of default mode network in adult, and they call it proto-default mode network. So the attempt to study the default mode network looking for recurrent spatial patterns already studied in adults seems to generate a discordant and confusing scenario. For this reason, we chose to concentrate our attention on a different fingerprints of the default mode network. So we will study, we will check the correlation and anti-correlation patterns that have been studied in adults. And these are characteristic of default mode network. So our probe won't cover the whole brain to study the extended network, but we will focus on the anti-correlation, in particular way, the anti-correlation between EFS, so intraparietal sulcus, and lateral parietal area. And we will try to use our 20 channels that is an adequate number to study only this kind of pattern to detect the anti-correlation intra and inter hemispheres and the correlation between homologous area. But all of this, provided that our probe is precise and correct. Indeed, the effective arrangement of sources and detector onto the scalp is critical to optimize the probe sensitivity. So usually, in particular way, in infant study, the probe is carried out in an intuitively way by placing approximately sources and detector onto the scalp. And this can lead to a suboptimal result. So for this reason, we are working on the development of a tool that is able to provide an anatomically reliable reference of the cortical region that we want to investigate, and a sensitivity map that we can consider as a cortical projection of the probe. In order to realize this tool, we started working on the most easy and general case, adult, and we tried to reproduce the previous studies by Cooper using an MRI atlas Colin 27, that is a high-resolution volumetric atlas of the brain, of the adult brain, and we realized a synthetic probe. In order to realize this virtual probe, we used the 10-5 system. That is a high-density system used in EEG for electrode positioning. And we obtained this probe. I just want to show how large is our probe. I hope that you can distinguish in red sources and in blue the detector. And once we have obtained the positions of sources and detectors, we could run the Monte Carlo simulation with an algorithm, with a specific algorithm, in order to understand how light spread in the brain. And we obtain the measurement profile sensitivity and the whole probe sensitivity. And once we complete the forward model, we started working on the inverse reconstruction model. So we generated synthetic time series in order to simulate two foci of activation. These activations are non-symmetric, are homogeneous, and in this way, with the activation, we simulated localized perceptual changes in optical properties. And with this simulation, we are able to understand how functional near signal changes when optical properties change. So we attempted the reconstruction of brain activation map from sensor space to source space. I just want to show you how our model works. So on the left, you can see a slice of the atlas with the original activation. And on the right, the slice with the reconstructed activation. So in this case, with this example, we have a correct reconstruction. You can see a little bit blurred reconstructed activation, but this is an intrinsic effect of the reconstruction that we can fix. But we have the reconstructed activation in the same area, in the same position with respect to the original one. But in the last case, in this case, we have obviously an incorrect, a wrong reconstruction. And so what did go wrong in this case? To understand the error, we need to consider the local sensitivity. So with the green area are indicated this set of channels that are in the neighborhood of the activation. And you can see that we have no channel exactly above the activation. So this is the proof of how important is the probe design and the probe positioning. And this is only the proof of how fundamental is our work on the development of this tool. And we just started the work on the newborn, and we used a simulated 4D head model that is a dedicated atlas that gives us information from 29 to 44 weeks post-menstrual age. And we are using the 4 weeks dedicated atlas because we are dealing with term infant. But in order to apply the same method that we developed for adult, we need to know the positions of probe and detectors. And so our challenge has been to map a planar geometry on a curved surface. And the geometry is the geometry of our probe. So 2 detectors and 8 sources for each hemisphere. So we have an hexagonal arrangement for each hemisphere, and the distance between source and detector should be of 2 centimeters. And we have to maintain this distance when we map this geometry. As you can see, we obtain the synthetic probe that should be a reliable representation of the probe that we will use in the actual measurement. You can see a symmetric probe that covers the area of interest. And we succeed in obtaining this mapping using Blender. This is a new tool, new for neuroscience. It is free software that is used to obtain animation, films, or video games. So once we have obtained the position, we run the Monte Carlo simulation, and we obtain sensitivity. In particular way, you can see the single channel sensitivity and the whole probe sensitivity. And a high sensitivity means that a high number of photons are passing through that particular point on their way to the detector. And you can observe how we can reach, with light, we can reach the cortical areas. But we are able to obtain a better representation of the probe sensitivity because we succeed in obtaining a cortical projection on the probe of the probe sensitivity. So we can see exactly which area we are studying. And if we compare with the target areas, we can say that it is a good result. And using the information of these from this tool, we developed the actual cap. This is a prototype of the cap. And I'm glad to announce that we reached a very important milestone for my project. Because a few days ago, we started our acquisition. We acquired the first 2 subjects and in the Rovereto hospital, because our lab has an important collaboration with the hospital. And for this reason, we have the opportunity to acquire directly in the nursery of the hospital. And the acquisition are made on sleeping in newborn of 1 to 3 days old, and are long-term acquisition. And this is a great advantage. Once we will obtain all the acquisition, we will be able to start with data analysis. We will study the newborn brain as a network characterized by nodes and edge, so functional links between the nodes. And our challenge will be to find the right parameters, the right constraint, in order to develop a model able to describe in the right way the process. So I'm looking forward to show you my result as soon as possible. And thank you to each member of the BrainNet Lab, and thank you for the attention.
Time for questions.
How much is reliable functional NIRS to detect the activity of deeper regions of the default mode network, for example, the precuneus?
Yes, this is a good question, thanks. But we are not able to detect that region of default mode network because it's too deep. So in our study, we chose to detect other fingerprints of default mode network because we want to use also the advantages of NIRS with respect to fMRI. So we cannot do a study of the extended network, and we are focusing on particular areas because we are not able to detect the deeper regions.
Okay, thank you.
Hi, thanks. This is really interesting. I didn't really understand how this is related to the search. Are you looking for anti-correlations? Is that what you're putting the sensors there? Because anti-correlations are part of the information you're looking for?
Yes.
And how does that play into this entire graph theoretical approach, which would suggest is typically applied within a network?
No, this is a future step, and with this we can consider also the correlation between different hemispheres, and we have the possibility also, because of the capability of the NIRS to realize long-term measurement, to study also the dynamical functional connectivity. And about the use of graph theoretical approach, yes, this is a future step, and we are thinking about how to realize this kind of model.
Is there work in the NIRS field about anti-correlations? Because in the neuroimaging field there has been a big debate on whether those are partially artifacts of global signal normalization or regression, and so these anti-correlations get reduced when people do not implement some pre-processing steps. So do you have any prior knowledge that you'll find them with NIRS?
No, to date this is maybe the first time that someone used anti-correlation pattern to study the connectivity, so I will
okay,
let you know.
Thanks.
Other questions? No? Okay, let's thank Mariagrazia. Thank you very much. Our last speaker for today is Joshua Zonca. He works with Giorgio Coricelli and Luca Polonio. He works in the Learning and Decision-Making Lab about strategic sophistication, attention, and cognition. Just started the second year too, and he has a background in cognitive neuroscience. I take the opportunity to remind you to vote at the end of the day, at the end of the poster session, and also at the end of the Joshua talk, we will have 5 minutes left for a short panel with all the speakers if you have more questions. So start to think about more questions for all the speakers.
Do you hear me? Hi everybody, I'm Joshua. I'm working with Luca and George in the Learning Decision-Making Lab. And today I want to tell you something about strategic sophistication in interactive contests. Basically, when we deal with interactive contests, we use game theory. Game theory is basically a model to represent every situation in which we have to make decisions, and how our decisions are influenced by the decision of other decision makers that are in the same environment as us. So, for example, imagine that you have to go on holiday and you have to choose between taking the highway or taking the panoramic road. If your aim is to reach your destination as fast as possible, of course, if you are in isolation, if you are a single decision maker, it will be easy to choose the highway as a choice, as a strategy. But of course, this is an interactive situation because you are not the only driver in the environment. You are not the only driver in the world that is going to that destination. And so it becomes an interactive situation in which you have to predict what are the possible choices of the other drivers, of the other decision makers in the environment. And so you can evaluate if it is worth to take the highway or the panoramic road given the possible choices of the other decision maker in the same environment. So in the laboratory, of course, we don't use cars and highways, but we use matrix games that are very simple representations of these types of interactive situations. So basically, a matrix game is a matrix like this that is shown to two participants that are playing at the same time in the laboratory. So for example, there are a row player and a column player. The row player has to choose between row 1 and row 2, while the column player has to choose between column 1 and column 2. Every possible combination of choices of the row player and the column player determine a cell, and every cell returns two outcomes, one for the row player and one for the column player. So the green one is for the row player and the red one is for the column player. So for example, if row player chooses row 1 and column player chooses column 2, this is the results of the game, and both participants get 4. Of course, here there are different strategies that can put in act. If you were the row player, for example, you may be attracted by this second row instead of the first row because the average payoff here is higher than here. 5 and 3 is higher than 3 and 4. But then you have to realize that it's a strategic situation, it's an interactive situation, and also the column player has to make the decision that can influence the outcome of your own decision. So for example, you might notice that the column player has a dominant choices, as is called in game theory. So the column 2 is better than column 1, whatever the row player will choose. And so probably the column player will choose column 2. Given that the column player will choose the column 2, for the row player is in his best interest to choose row 1 instead of row 2 because 4 is higher than 3. Now in the laboratory we observe heterogeneous behavior of participants, and not many participants actually show strategic behavior. Why? There are at least 2 possible explanations for this, or at least 2 classes of explanation. One is a social type of explanation. So we behave differently. I'm a strategic player and you don't, simply because we have different prediction about what other players will play and how other players will behave in this environment. A second type of hypothesis is a cognitive explanation. So differences in level of sophistication depend on the implementation of some cognitive process. So the strategic player probably is using some cognitive process that allow him to use the strategic component and solve the task in a strategic sense. Now, to this sense, last year, Luca and Giorgio came out with this very interesting paper, a very interesting idea to test games with eye tracking. So to analyze which are the high patterns that really underlie the process that players are putting in act in order to solve this game. And what they found that basically the analysis that participants did in analyzing the game is a very strong predictor of the choice. So the way in which you analyze the game predict if you will be strategic or not. Now this suggests us that probably there is something really procedural in the way a game is analyzed and is related with how we acquire and integrate the information and the relations that there are in the payoffs in these metrics. And so our hypothesis in this study is to really test that strategic sophistication roots in something that is not specifically social, but is something related to the ability to acquire and integrate information in whatever environment, also in non-interactive settings. So we have 2 main hypotheses. One is that we will be able to predict strategic sophistication while analyzing the ability of participants to acquire and integrate information and to look for the relevant information in the environment and disregard the irrelevant information in a non-interactive context. And as this player can use a proactive cognitive control approach in order to being able to know in advance which are the information that they want to look for and disregard all the irrelevant information they can encounter in the environment. Our approach, so in this study, is to use 3 different eye tracking tasks. So it's a within subject design with, in which we tested 57 participants that perform all of these 3 tasks. The first task is an interactive task, so are the games that I showed you before, while the second and the third task are non-interactive tasks, are individual tasks. The first one, the second one is a reasoning task that aims to disentangle between abilities in relational information acquisition and representation, while the third task is a very simple visual source task that can give us, tell us something about proactive and reactive cognitive control. Our approach will be to individuate the parameters, attentional parameters that can explain behavior in this non-interactive context and see if those parameters can predict how these players will choose in games when they are faced with an interactive context. First task, the games. exactly the games that I showed you before. So a matrix game, how we conduct the analysis. Basically, we identify some regions of interest that are placed basically around the payoffs of the matrix, and then we analyze the transitions, the high patterns that connect these regions, these symbols. We have three main categories of transitions. The first one is transition connecting on payoff, the on payoff of the participant, if it is a role player. Transition connecting the other player payoff, and inter-cell comparisons. Basically, we took the proportion of each of these type of transitions, and based on these three parameters, we used a mixture model cluster analysis that, without imposing any requirement about number of clusters and so on, returning the best clusterization that maximized the Bayesian information criterion. Basically, the cluster analysis returned three groups of players in terms of analysis of the game. The first group, the yellow one, is level 1 players. Level 1 players are the non-strategic ones, are the players that just look at their own payoff and they disregard the information about the other player. Level 2 players, the blue one, are the strategic one, are the players that analyze their own payoff and do the transition between the other payoff, and they integrate this information in order to make a strategic decision that takes in account the possible moves of the other player. Then there is the third group. This third group is focused on inter-cell comparison. Why they are doing this? Because there are two types of players in terms of behavior and social preferences. So there are cooperative players that want to maximize the joint payoff to the participants. Basically, they want to be nice also with the other participant. They want to gain money, but want also the other opponent to gain money. While the competitive players are not so nice because they want to gain money, but they want to maximize the difference between their own gain and the gain of the opponent. So they want the opponent to not gaining money, basically. Okay, keep in mind this. We have 4 types of players that analyze the game in a different way, and play the game in a different way in a strategic context. Now, second task, the reasoning task. This is a completely individual task. It's a reasoning task in which we have conditional relations that can be arranged in a different way, and that can be linked by transitive relation. As you see here, there is a transitive relation that connects these two symbols and a transitive relation that connects these two symbols. Participants can see this screen for 9 seconds, then this screen disappears. Then we show this screen that tells the participant which is the true state. Of course, before the participant didn't know if the square was true, if the circle was true, was only aware of the conditional relations that were connecting these symbols. Once we tell the participant which is the true state, the participant has to do some inferences. For example, if square is true, as we said, circle is true. And if circle is true, triangle is true. If triangle is true, cross is true. So the participant has to answer with all the symbols that are necessarily true given the true state. So in this case, has to answer all the 3 symbols that are in the screen. Of course, the participants cannot know in advance which are the answer, because he has to see which is the true state in order to respond. But this is important because in this phase, in the encoding phase, the phase in which I'll be focused on the analysis, we are going to target the ability to acquire relational information and acquire all the relevant relations that can be used in the following stage to do inference, whatever is the true state that I'm giving to the participants. We have some hypotheses about the optimal behavior in this task. So we expect participants to do an exploratory phase at the beginning. So they have to understand which are the relations in the current trial, basically, because they don't know, because every trial has a different structure and different relations. While in the second phase, in the consolidation phase, so the second part of the coding phase, they should have understood which are the relations in the environment and should focus only on relevant relation in the current structure. And so with the iData, we should observe that they really focused on relevant transition and disregard irrelevant transition. How we separate between exploration and consolidation phase? Basically, we just used the fact that we observe an increase in average fixation duration in our participant at a certain point of their encoding phase in average. So basically, for each participant, we individuate the moment in which they start to significantly increase the average fixation duration. And based on this, we separate for each participant the exploration phase and the consolidation phase. The average group threshold is around 3.2 seconds out of 9 seconds of the encoding phase. How we did the analysis? Again, we created some region of interest that around the symbols, and we analyzed fixation and transition connecting this fixation in this region of interest. We created some attentional indices that tell us something about the type of analysis that they are using to acquire this information and to encode this relational information. I want just to focus on one important parameter that we used, that is relevance, logically relevance of relations. So basically, trial by trial, we individuated all those transitions that are connecting symbols that express a meaningful relation in terms of logical properties. For example, if-then relation, or relation, and transitive relation. What we expect that participants that perform the task optimally in the second phase, in the consolidation phase, have understood the structure of the problem, and they start to focus on only on this relevant transition because they understood the relational space in which they inserted in. So again, we took all these indices that explain behavior in terms of acquisition and information, and we clusterized participants based on this. The cluster analysis returned two groups of participants that, in a not politically correct way, I called high reasoning participants and low reasoning participants. So the high reasoning group basically is performing the task optimally and is doing the analysis that we expected for our optimal participant. So is searching for relevant relation in the first phase and is consolidating only this relevant relation in the consolidation phase. While the low reasoning group is not really understanding which are the relations in this, all the relations in this space. For example, it doesn't see that there is a transitivity relation that connects these four symbols, and then in consolidation phase, they just consolidate in working memory what they acquired in the exploration phase. That is a not exhaustive representation of the full relational space they are. This is just to show you that, interestingly, the two clusters are very different in terms of performance. So the high reasoning group is indeed more accurate and faster in solving the task. And one interesting aspect is that we control for the working memory component. So we wanted to control that this is not just due to memory capacity, because maybe the reasoning group is just more memory and is just better in memorizing symbol. But we have 4 different measures of working memory, and none of these return a significant difference between the two groups. Now, we have 2 groups of participants defined based on the analysis they did in the reasoning task. That is a non-social task. Then we compare these 2 groups on game playing, on interactive game playing. What we observe, very simply, is that the high reasoning group has a higher proportion of strategic choice compared to the low reasoning group. And here on the right, we are going to look the distribution of each type of player across the 2 reasoning groups. The first thing that is very clear is that the level 2 players, that are the strategic ones, and the level 1 players, that are the non-strategic ones, really are distributed in a different way between the 2 groups. So more than 70% of the high reasoning group, the level 2 players, the strategic players, are in the high reasoning group, while more than 70% of the non-strategic players are in the low reasoning group. Now, you can notice that there is not a clear pattern instead for cooperative and competitive people. So these types of player do not seem to show a difference in terms of abilities in acquisition of information and integration of this information. What we reason is that, by definition, these players can have social preferences because they are cooperative and they are competitive. And so this ability in solving the game in the sense of acquire the information in the right order and in the right way can be hidden by the fact that a strategic player that wants to be nice with the other participant just wants to be nice with the participant, and even if he can solve the game, wants to maximize the payoff between him and the opponent. So what we reason is that if we can rule out the social component for a moment, we should observe, if our hypothesis is true, an increase in strategic choices in this group because they are able, in principle, to acquire the information of a game in a good way, while this group that we think is not able to do it should not show an increase in strategic choices. And this is what we observe. Basically, what we did is recalling all these participants, and we make them play with a computer instead of a human player. So here the social component, or at least the social preference component, the motive to help the other should be ruled out, because it's nonsense to want the computer to gain money or something like this. And what we observe is that only the high reasoning group show a significant increase in the percentage of strategic choices. Last task, the visual search task. It's a very simple visual search task that we always see in the literature. So there is a target, and there are a bunch of distractors. You have to say if the target is present or not as fast as possible. Very easy, very simple. The analysis was carried on by dividing again the screen in region of interest and analyzing saccades and transition and fixations across this region of interest. We have two hypotheses of two possible strategies that participants can use in this type of context. One is a proactive strategy. So the participants select in advance a pattern of visual information acquisition and repeat this for every trial, independently of the position of the distractor. The second type of player that we want to find is reactive player that just follow the position of the distractors in the environment, and they don't select in advance a strategy. And so every trial for them is different, and we should observe a different analysis every trial by trial. And this is what we found. We again did our cluster analysis, in which we use these parameters of attention in order to clusterize participants in terms of analysis in this task. And what we found is two groups. A group that is doing a linear scan path and is repeating this trial by trial. Basically, they want to find the target, and so they are doing the same thing trial by trial, and they stop when they found the target. The second group instead is completely attracted by the distractor. So it doesn't select a strategy or scan path in advance. They just look at the screen in a random way, or at least they follow the distractor and the configuration of the symbol in order to find the target. What we observe in terms of behavior here is that this group is more accurate, but is lower in this task. Now, again, we took these two groups and we compare the strategic behavior, so behavior in games, in interactive context. And very simply, what we found is that the percentage of strategic choices in the proactive group, so the group that select a strategy in advance in order to solve the visual search task, is higher than the reactive group in game playing, basically. Conclusions: we observe an optimal encoding of relational information in a reasoning task, and so we can infer in some way that sophisticated analysis in interactive contexts can be linked and should be linked by this ability in acquiring and integrating the relational information that are important in a game, in a strategic setting. Then we saw that a proactive strategies in a very simple visual search task, in a perceptual task, can tell us something about the strategic behavior of the participant. And that's why we think that in games it's really important that you select in advance which are the relevant relation that you have to look for and the relevant comparison between payoff that you have to do in order to solve the task in a strategic way. So the take-home message is that strategic sophistication is not simply a social skill. It's not just that we are different in terms of strategic sophistication because we think differently about the other players, but there is really something in the way in which we acquire and integrate information in order to form a complex representation of this interactive situation. Thank you.
Thank you very much, Joshua. You can stay here, and I ask you, the other three speakers, to come in. So if you have a question for Joshua or for all the others, please don't hesitate. Please.
Thank you, very interesting. I have two questions. The first one is, I noticed that you measure Raven matrices performance.
Yeah.
Isn't that entirely already sufficient to capture the difference in the inter-individual differences in the rest of your tasks?
It is related, and actually it's related with the score in the Raven is related with the reasoning task, but it's actually also related with the visual search task. So it's really something about fluid intelligence and attention and whatever. First, we wanted to analyze really eye movements, and so patterns of information acquisition and integration, and not only the score in a reasoning task, because we already know in the literature that there are differences in cognitive abilities between strategic and non-strategic players. In the Raven, is quite difficult, also if you put it in the iLink, to really understand which is the strategy they are employing in order to chunk the information in a meaningful way in order to find the relation and the rule that is in the current trial. And so we wanted to create a task that we could control in terms of which relation are in what position and the type of relations that are in the environment, and so we can control with eye tracking analysis the ability to acquire and integrate this type of information in a meaningful way.
My second question relates to, there is an aspect that I found really interesting, that is, you can be highly strategic and a high performer, and you can solve the task to your convenience if you want, but you do not necessarily do it. And so the behavior that you actually implement is not necessarily the consequences of your ability to analyze the problem and come up with a solution, which I think is an interesting aspect that is not often emphasized. I mean, yes, I think you would agree that you can capture many of these aspects already with general indices like fluid intelligence indices, like the matrix, Raven matrices. You can describe even more if you add further tasks like you have done. But I think it's quite interesting that in the end, the fact that you can measure these abilities is then, whether you implement them or not, is hugely, highly dependent apparently on the way you socially read the situation.
Yeah, this is interesting in economics, because people that do game theory usually will think that these people are hassles. Like, economists just can see the fact that you don't play optimally, and you don't reach the equilibrium of the game, and so you're stupid or whatever. But yeah, there is really a intrinsic social component in a game that that must be integrated in the, of course, cognitive process that is behind this game, and then both the components are necessary, because if there is this social component in a non-cognitive sense is not present, we just don't need games, because we just need cognitive task or some reasoning task, and we'll measure abilities of decision making, basically.
Other questions?
Thank you, Joshua. I have a question for you. So my general question is, how do you think these group profiles are stable and consistent? I mean, you show that across different tasks, the strategic one is still more strategic and the non-strategic is non-strategic. Well, let's suppose, for example, that you push the motivation, okay, to maximize the gain on each task and the task that you, okay? What do you predict you predict that, for example, a shift in a strategic group from one to another, and if so, this strategic may be more non-strategic in the sense that they will look for more for themselves, or maybe the non-strategic will shift in the strategic, meaning that they have to maximize in relation to the other one.
This is very interesting because it's actually the final goal of my PhD. So understanding, before understanding which are the processes that strategic players are employing, but then understanding how we can trigger sophistication in participants that are not employing this strategic behavior. And so to me, there is the possibility, of course, to increase motivation. Keep in mind that here the motivation is already quite high because participants are imposed based on performance, and so it's really an economic setting that tries to maximize the motivation. I would hypothesize that there will be a slight increase in a motivational sense for both groups. And I think that for the non-strategic group, it must be triggered something more procedural in the way they acquire and they think about the problem in order to be strategic. I'm not saying for sure that these people cannot do it. I'm sure that almost all of these participants that I have in my study can be strategic, but of course they have to use a way of thinking that goes beyond the heuristics that they're using in the task. And an interesting question is to find which is the process that must be triggered in order to make these people start to think strategically, because this is the goal of this, that people can think strategically.
Just to follow up on this question, I was wondering, like here we're talking about sophistication, but then how about switching between maximizing strategy and like social preference strategy? So like, for example, moving from a cooperative to level 2 strategy. Do you think this is actually more like flexible, or do you think like this switching is as difficult as moving from one level of strategy to another?
I think it's easier in those participants they can that already analyze the game in a proper way in strategic sense, but they decide to cooperate because of social preference, because those people already know how to analyze this game and know what is the solution that is in their best interest, but they don't do it because they want to be nice with the participants and so on. Of course, it will be more difficult for a level 1 player, so a non-strategic players, to move to a strategic level and move to level 2 players, but I think this can be done for sure. And I think in the literature there are some evidence that we can increase sophistication by priming with some task before, but we really don't understand how this is possible and which cognitive process we have to use in order to become strategically analyze a game like this in a strategic way.
Other question, curiosity, technical, I don't know. You can also ask them how they are so good in staying time and it's a good skill to acquire during your PhD. While you think about questions? Okay, Chiara. Well done.
Okay, so my question is for Mariagrazia, actually. So you showed two different, completely different results from previous studies. So some of them found the default mode network, some of them didn't find it. Which is your expectation? Do you have any prediction? Do you expect to either find it or not find it, or do you expect to find some nodes? And I don't know. I mean.
You have to consider. Okay. You have to consider that the previous study that I showed focuses on the focus on the spatial pattern of the full mode network. And so we have discordant results. We are examining for the first time, because there are no studies on patterns of correlations and anti-correlation as a fingerprint of the full mode network. So this could be the first time. So we don't have any other studies that we compare with our work. So it is the first time that at the full mode network that this particular fingerprint is used to detect the emergence of the full mode network, in particular way in infant. We are working on an acquisition of adult in fMRI with functional nears. In this way, we will have a first approach of this anti-correlation pattern with that it is visible with fMRI with functional nears in adult. And my result will be the first result with this kind of approach.
No more question, curiosity? So I take the chance to acknowledge and present you the team that organized this year, the Doctoral Student Day and the Welcome Day, who are Eugenia Gianni, Chiara Maffei, Poppy Sharp, Francesca Perna, and Flora Nerpic, under the supervision of Professor Veronica Mazza. I also take the opportunity to thank all our speakers and to invite you to enjoy this day and to have a wonderful year here at CIMeC. Thank you very much.